In a remarkable breakthrough that could redefine the future of cancer treatment, scientists have unlocked the complete biosynthetic pathway of one of the most powerful and widely used chemotherapy drugs, Taxol. This achievement marks a pivotal moment in pharmaceutical science, offering a sustainable, cost-effective, and scalable solution to produce a drug that has long been shrouded in complexity and expense. For decades, Taxol has been hailed as a cornerstone in the fight against some of the deadliest forms of cancer. Its ability to halt the growth of malignant cells by interfering with their division has made it an indispensable tool in oncology. However, its production has always been a challenge. The traditional method involves extracting precursor compounds from the bark of the Pacific yew tree, a slow-growing and endangered species. These precursors are then subjected to a series of complex chemical transformations to yield the final drug. This process is not only costly but also environmentally damaging, requiring large quantities of solvents and energy-intensive procedures that leave a heavy carbon footprint. The scientific community has long sought a better way. For over 30 years, researchers around the world have been piecing together the biological puzzle of how Taxol is naturally produced within the plant itself. Understanding this natural process was key to developing a more efficient and sustainable alternative. Now, after years of dedicated research and collaboration, a major piece of the puzzle has finally fallen into place. At the heart of this discovery lies the identification of two crucial enzymes that were previously missing from the known biosynthetic pathway of Taxol. These enzymes play a vital role in the final stages of the compound's formation, converting inactive precursors into the biologically active form of the drug. With these last pieces of the molecular jigsaw now understood, scientists can replicate the entire process in a controlled environment, specifically, inside genetically engineered yeast cells. This innovation opens the door to a revolutionary approach in pharmaceutical manufacturing. By transferring the genetic instructions responsible for Taxol synthesis from the yew tree into yeast, researchers have effectively created microscopic factories capable of producing the drug on demand. These yeast cells act as living bioreactors, following the same biochemical steps that occur naturally in the tree, but under carefully optimized conditions that maximize yield and purity. The implications of this development are profound. Not only does it promise to significantly reduce the cost of producing Taxol, but it also eliminates the need to harvest rare and ecologically sensitive trees. Instead, the raw materials required for the process come from more readily available sources, such as yew needles, a byproduct of pruning or forest management that would otherwise go unused. This shift not only preserves biodiversity but also aligns with global efforts to promote greener and more circular approaches to resource use. One of the most exciting aspects of this new method is its scalability. Unlike the traditional semi-synthetic route, which requires extensive purification steps and precise chemical reactions at each stage, the biological approach allows for continuous production using renewable resources. Yeast cultures can be grown in large fermentation tanks, much like those used in brewing or biofuel production, enabling the manufacture of Taxol at industrial scales without compromising quality or safety. Moreover, the environmental benefits extend beyond the reduction in chemical waste. The entire process avoids the use of toxic solvents and reagents that are commonly employed in conventional chemical synthesis. These substances often pose risks to both human health and the environment, contributing to pollution and long-term ecological damage. By replacing them with a clean, biological system, this new method represents a significant step forward in sustainable chemistry. From a public health perspective, the potential impact of this discovery is equally transformative. Taxol is among the most effective treatments for several types of cancer, yet its high cost has limited access for many patients, particularly in low and middle-income countries where healthcare resources are already stretched thin. In regions where the burden of disease is growing rapidly, especially with rising rates of ovarian cancer, the availability of affordable Taxol could mean the difference between life and death for countless individuals. The current price of Taxol remains prohibitively expensive, with the active ingredient costing tens of thousands of dollars per kilogram. This exorbitant cost is largely due to the inefficiencies of the existing production methods. By streamlining the process and eliminating the need for costly intermediates and laborious purification steps, the new biosynthetic approach is expected to cut the overall cost in half once fully optimized. Such a reduction would make it possible for more hospitals and clinics to stock the drug, ensuring that it reaches those who need it most without delay. Beyond the immediate economic and medical benefits, this breakthrough also sets a precedent for the future of drug development. It demonstrates the power of combining cutting-edge genetic engineering with a deep understanding of natural biological systems. As scientists continue to explore the vast potential of microbial cell factories, we may see similar innovations applied to other complex molecules currently reliant on inefficient or unsustainable production methods. The journey to unlock Taxol's biosynthesis has been a testament to perseverance and interdisciplinary collaboration. Researchers have drawn upon diverse fields, including plant biology, biochemistry, genetics, and synthetic biology, to unravel the intricate web of chemical reactions that give rise to this vital compound. Each step along the way has built upon previous discoveries, gradually assembling a comprehensive picture of how nature constructs one of its most potent anti-cancer agents. Now that the full pathway is understood, the next phase involves refining the yeast-based production system to achieve even greater efficiency. Scientists are working to optimize the expression of the necessary genes, enhance the metabolic flux through the desired pathways, and minimize the formation of unwanted byproducts. These improvements will ensure that the final product meets the stringent quality standards required for pharmaceutical use while maximizing output and minimizing waste. The team behind this discovery is also focused on translating the research into real-world applications. A patent has already been filed to protect the technology, and plans are underway to establish a spin-off company that will commercialize the process. This move underscores the importance of bridging the gap between academic research and industry, ensuring that scientific advancements can be harnessed to benefit society at large. As the project moves toward commercialization, it will be essential to engage with stakeholders across the healthcare ecosystem, including regulatory bodies, manufacturers, and patient advocacy groups. Collaborative efforts will be needed to navigate the complexities of bringing a new production method to market, from obtaining approval for the drug to negotiating pricing structures that balance affordability with sustainability. Looking ahead, the success of this initiative could inspire a broader shift in how we think about drug production. Rather than relying on traditional chemical synthesis or resource-intensive extraction methods, the future may belong to biological systems that can replicate the elegant efficiency of natural processes. This vision aligns with the growing emphasis on green chemistry and sustainable innovation, reflecting a commitment to addressing both global health challenges and environmental concerns. The story of Taxol's biosynthesis is not just about science, it is about people. Behind every breakthrough in medicine lies a deeper motivation, the desire to improve lives and alleviate suffering. For the millions of women affected by ovarian cancer, and for all those battling other forms of the disease, this new method offers hope. It represents a tangible step toward a future where life-saving treatments are no longer a luxury reserved for the privileged few but a right that can be extended to all. As the research continues to evolve, it serves as a powerful reminder of what can be achieved when curiosity, determination, and collaboration converge. The road to unlocking Taxol's secrets has been long and arduous, but the rewards are far-reaching. By harnessing the power of nature and the ingenuity of modern science, we are not only advancing our understanding of biology but also paving the way for a healthier, more equitable world. This groundbreaking discovery marks a turning point in the fight against cancer and the future of pharmaceutical production. For decades, scientists have worked tirelessly to understand how nature creates one of its most powerful anti-cancer compounds, Taxol. Now, with the full biosynthetic pathway unlocked, we're on the verge of a new era where this life-saving drug can be made more efficiently, sustainably, and affordably. By harnessing the power of yeast as a living factory, researchers have developed a method that not only cuts costs but also eliminates the need for harmful chemicals and endangered plant species. This means less environmental impact and a more ethical approach to medicine production. It's a win for patients, doctors, and the planet alike. As this technology moves toward commercialization, the hope is that it will make Taxol accessible to those who need it most, especially in regions where rising cancer rates and limited healthcare resources make treatment a distant dream. Lower prices, combined with greener methods, could change the landscape of global oncology care. So what does this mean for the future? It means innovation is no longer just about creating better drugs, it's about making them available to everyone, without compromise. It's about using science to build a healthier world, one breakthrough at a time. If you found this story inspiring or want to stay updated on the latest in medical science and sustainability, don't forget to like, subscribe, and hit that bell icon. Thanks for watching.